We're live.
All right, welcome back, everybody. This is our second talk of our afternoon session. Today, we're going to hear from Dr. Pouya Bashivan. Pouya is a new professor at McGill University, so we're super happy that he joined the corpus of faculty members here in Montreal. He did a brief postdoc here with Professor Irina Risch at Mila. But before that, he completed a postdoc with Professor Jim DiCarlo at MIT and received a PhD in computer engineering from the University of Memphis. Generally, Pouya seeks to develop models and algorithms that can explain, predict, and ultimately regulate the brain responses during visual tasks, especially ones that require short and long-term memory. His research builds on top of various tools and theories developed in machine learning, neuroscience, and cognitive science. So very much in vogue with the theme of this conference. And we're very happy for you to join us, Pouya. And your talk today is going to is titled "Computational Models of Visual Perception for Prediction and Control." And the floor is yours.
Thanks, John. Thank you for the beautiful introduction. Yes, this is Pouya Bashivan, so I'm going to give a talk. It's a short talk about some computational modeling that I've been doing. This is mostly my postdoc work in Jim DiCarlo's lab, and it's about making models for prediction and control. So without further ado, I'm sure that most of you here care about at least two things. You most probably care about AI and neuroscience. Maybe you also care about Montreal, and that will make you the perfect fan of the main conference. But even if you're not a big fan of Montreal, still just caring about AI and neuroscience would be just enough for the sake of this talk. For many decades, these two fields, the AI and neuroscience, they've had this symbiotic relationship between each other, informing each other, basically. And my research is also kind of at the intersection of the two, as you will see. But the focus of this talk today is mostly about how we can use AI tools or AI in general to kind of elevate our understanding of how the brain works. And most of the material that I will be presenting to you today is going to be focused on the part of the cortex that is called the ventral pathway, is hypothetically is used for object recognition in the brain. But the methodologies and all the approach that I'll be talking about is more general, and it could, well, we hope that it could be adapted to other areas, other regions, or even other species. So the behavior, so let me tell you a bit of background about what I want to discuss today. My background is in control engineering, so I did a bachelor and master's degree. Could be adapted to other areas. I'm hearing the echo of myself, but so the behavior. So let me tell you a bit of background about what I want. So and as a control engineer, what you usually have in a control problem is a controller and a system. And the system usually accepts a set of inputs and it produces a set of outputs. And the job of the controller is to basically devise a control signal given a desired output that, as a system designer, you have in mind. And the core of what I want to discuss today is to basically relate this framework to a system, which is visual system in this case, and the predictive models of this system, which in our case, that would be the artificial neural networks. And the core question that we want to discuss today is, can we use predictive models to control the neural response patterns in the brain? All right, so of course, we were not the first people thinking about this, basically to control the neural responses in the brain. There's been many different approaches that have been proposed throughout the years. We've had electrical and magnetic kind of stimulation that goes back to one of the early studies by Jose Delgado in 1963 that he basically implanted a bull, an electrode in the bull, basically going to the caudate area in the brain, and using a remote device, he showed that he could, in a way, he could control the behavior of the bull, which in this case was a charging bull would just come to a stop by the desire of the experimenter. And another good example is deep brain stimulation, which is one of the very good applications of how neuroscience could be used in applications that are life-changing. And we also have pharmacological and chemogenetics methods like Musimo and Gretz. And also we have, more recently, we have the optogenetics that have been really, really helpful and really revealing in neuroscience during the past several years. One thing that is common between all of these different methods is that usually these methods are designed in a way that your ultimate goal is to control the behavior, and your tool is through controlling of the neural states. So you're basically manipulating the neural responses in order to have control over the behavior. And in all cases, they are considered invasive methods, and you need direct access to the neurons. And control is also as precise as the stimulation method, meaning that the level of preciseness that you have in order to manipulate the neural responses, that would determine how much control you have over the behavior or the neuron. But what I want to talk to you about today is kind of a bit different. What we want to see here is, can we control the brain's activity through sensory stimulation? And so the behavior that we want to talk about today is visual object recognition. And in general, primates, and more specifically humans, are very good at recognizing different objects. We can recognize among many thousands of objects, and we can do so regardless of the viewpoint of those objects, and it only takes us tens of milliseconds to accomplish this. And as I said earlier, the part of the brain that supports this behavior is called the ventral visual pathway, and it consists of a series of visual cortices that starts from area V1 and ends in an area called inferotemporal cortex, or IT for short. And many decades of neuroscience, it has shown us that the neurons, for example, in V1 cortex, they respond best to oriented bars. And as you go deeper, you get to V2 area, which the best description so far for what these neurons do is the correlated patterns of V1 responses. And then you go deeper in V4, you get the neurons that respond to complex curvatures and gratings. And as you get to the tip of this IT cortex, specifically in anterior IT cortex, these neurons and the visual neurons, they begin to respond really highly to complete objects or like complex shapes that look like specific objects. And one thing to note here is that as we're getting away from the retina and from where the light comes in, the input to the system, it's becoming harder and harder to understand what the neurons are actually doing, what aspects of the visual world they're capturing, by trying to figure out what kind of stimuli they best respond to. Before we get into the experiment and the actual research that I want to talk about, let me tell you a bit about how the data is collected in this case. So the data, most of the data that I'm going to talk about is electrophysiological data that is recorded from macaque monkeys using these tiny electrodes. They're called beta-rays. And what is usually done is that one or several of these electrodes are implanted on the surface of the cortex. And as the monkey, as the animal, views these different objects, we basically record the responses in the sense of spiking rates from these individual sites that could be one or multiple neurons. And to give you a better idea, so the kind of data that we're recording, it kind of looks like something like this. So each column here, I'm showing a different image. And here, I hope that you're seeing the cursor. So we have site 1, which is one of the electrodes in this case. And you see that as this monkey is seeing each of these images, you're basically capturing different levels of activity in the sense of like how many spikes that the specific neuron is making in response to each of those images. And because this data is inherently noisy, we tend to repeat the same images multiple times to get a better quality data, which is basically each row here it corresponds to one trial of showing this one image. And then we can repeat this for different sites. And of course, the number or the measure that we really care about in this case is the average spiking rate, which each of these sites are making in response to each of these images. And from this point on, we can go and now extend this to many neurons that we're recording, if you remember from those Utah rays, for example, each one of them had 96 electrodes. So we can put all of those numbers, the spiking rates, alongside each other. And we create these vectors of responses of neurons to each individual image. And we can now extend this more to having many such images. So in a sense, we're getting close to having a matrix-shaped data format like this one, which in this case, each row would correspond to one neuron, each column corresponds to one image. And the goal of our modeling project or our computational model, what we want to have is to be able to predict these pattern of responses that these neurons are generating. Yeah. So now that I told you a bit about where the data is coming from, let's get into like where the models are coming from. So most of the models that I will be talking to you about today, they're coming from artificial neural networks that they really caught on after this phenomenal paper in 2012 by Jeffrey Hinton's group, that they successfully trained a large, or with respect to today's standards, a relatively large neural network to do object recognition from pixels. And there's been many different works in the past years that extended this idea and this ideology. And right now we have these convolutional neural networks basically that are the current best predictive models of the neural responses in the brain. But one thing that I haven't told you yet is that now that we have the CNN model and we're recording from the brain, how do we actually compare these two with each other? So the process is very simple. We bring a set of images and we show the images to the animal as we're recording the brain responses, the firing rate or the spiking rates. And at the same time, we can show the same set of images to our CNN model. And because we have access to the internals of this model, we can look at the activations inside each layer or after the output of each of the layers in the neural network. And then what we really want to do is to kind of compare how similar the activations of those units in the model are to the neurons that we're recording from the brain. And in reality, instead of looking at individual units, we usually find this combination of units inside a layer that gives us the best approximation of what each individual neuron in the brain might be doing. And this approach was basically tried in recent years. And the finding was that this class of models are actually quite good at predicting the neural responses to individual images in some mid and high level cortices along the ventral pathway, including the V4 and IT cortex, if you remember from the figure. Now that I told you about these predictive models, let's go back again to the original question that we wanted to, that we started with. And that was the the goal of control. Can we control the neural responses using such predictive models? And by control, what I mean is basically to use a model to, in a generative sense, to synthesize new visual stimuli that would drive the neural responses, the population of responses, into a desired state. So in a way, we're kind of, the goal here is to control the state of this system, not the output. And so we tested this in two setups that we kind of thought of as like an exemplary cases. One case that we called maximal drive or stretch was the following. So we knew that when we show naturalistic images to these animals, we can record the responses, and if you imagine the responses, the distribution of those responses for one neuron, it will probably look like a kind of like a Gaussian, like this blue Gaussian plot here. So the goal of this experiment was to use this model to synthesize these images such that when we show these images back to the animal, it would actually cause the responses to be much higher than what we had for those natural images. And the process is fairly simple. We had 4 steps. The first step is to train this model, of course, the CNN, on object recognition tasks. This is done on an ImageNet dataset, and then once that's done, we fix the parameters, and then we use our neural data in a cross-validated way to create this mapping from those features inside the CNN to these neurons that we're recording from, in this case in the V4 area. And once we have that, we have this fully differentiable model that goes from pixels and gives us predictions of V4 neuron responses. And because this is a differentiable function, we can use it in a kind of reverse mode to now synthesize visual stimuli that the model would predict to cause high responses in those neurons, right? And the last step is to just go back and show those images to the animal, just to test how good or how accurate our control has been. So we did that. The first thing that I'm going to show you is how good the predictions of this model could be. So this is a scatter plot of different natural images, their predicted values on x-axis, their measured responses on y-axis. You see that the model is doing a quite good job. And if I show you what this specific neuron is seeing inside this receptive field, this is kind of looks like this part of the image that you're seeing here. This is the pattern that this particular neuron is seeing in V4. And you see that it's very complex to describe what kind of pattern this neuron was actually responding to. Now, if you show these synthetic images that we generated through the synthesis procedure, you see that for this particular neuron, we have achieved our goal in the sense that we could drive the responses of these neurons higher than the distribution that the natural images would generate. But at the same time, we see that the error in predictions for these set of images are still higher. And just to give you a better sense of like what are these images, when we look at these images, we see that they kind of have these complex curved patterns. And so let me tell you what these 5 dots are. These 5 dots are basically repetition of the same experiment using different random seeds. And you see that the output of this experiment for the 5 seeds, they're fairly very close to each other perceptually, but at the level of pixels, they're not. And we also evaluated how much success we had by looking at how much the extra firing rate we've caused by using this method, and we showed that on average it's about 40%. And so this was the first experiment. The second experiment, we looked at another case, which we called one-hot population. In this new experiment, it was kind of a bit more complex. We wanted to control a population of responses that we were measuring. And then in that case, we were targeting one specific neuron, for example, N1 here. And we wanted to drive the N1 responses really high, or as high as we could, while controlling the responses of other measured neural responses to be relatively low. And then we changed the target to different neurons that we were recording from. Before I show you what the synthetic responses, the responses to synthetic images look like, This is kind of when we did the experiment on natural images, and we tried to find the best natural image in our cohort, in our data set that would drive this target neuron that you see in dark blue. We tried to really drive this neuron, or find the image that drives this neuron high while driving the other neurons really low. And you see that this is, for this particular neuron, it's actually not really successful. And the goal of the experiment was kind of creating this pattern in red here. And when we did that, when we did the synthesis using the model, and we generated these images, we got this image on the right. And when we showed it to the monkey, we kind of got this pattern of response that, as you can see, it's much closer to with our desired response, which was like drive target and suppress everything else. And small note that I wanted to have here is that this is not meant to be like this experiment is not meant to be like a comprehensive experiment, but this was kind of only a proof of concept. So the idea was to formalize the desired response that we might want out of our neural responses, and then formalize it into something like an objective function or a loss function that would help us to achieve the control objective by optimizing that loss function. And of course, you can also go ahead and devise different types of objective functions for different control goals. And another experiment that we did is kind of like it's a tiny control experiment that we did for neurons that had highly overlapping receptor fields, just to make sure that our control experiment is not only finding neurons that have non-overlapping receptor fields. And again, we repeated that, and we found out that in most cases, we could achieve a relatively high level of independent control. So that was the first project. So what's next? Of course, there are different things that we could have done, but one thing that we decided might be interesting to proceed with was to ask whether we can apply the same techniques to measurement from the human brain. And this work was led by Ratan Moti, who's a postdoc at Nancy Camish's lab. He's actually wrapping up his postdoc currently, and soon he'll be looking for a position, I assume. And this was in collaboration with Jim DiCarlo and Nancy Camish. And the setup is pretty similar. We wanted to do basically the same thing with the human brain responses, but in this case, because we needed to do it in a non-invasive way, we used fMRI. The idea is the same thing. We have humans in the MRI machine. We show these images to these human subjects as we're recording their brain responses. And again, we have the same class of neural network models, convolutional neural networks, and we get the responses. We create this linear mapping to predict those responses. And maybe one difference here is that instead of looking at the specific neurons, because we cannot do that, here we looked at specific regions that you will see later on. We specifically looked at 3 regions: FFA, EBA, and PPA. Just maybe the overview of what the model could achieve only on a specific region, the phase-selected region, FFA in this case. So we fine-tuned the model, and we asked the model to make predictions on every one of these images that we had. We had about 200 images, and the process is cross-validated, and the images were randomly randomized and then cross-validated to make the mapping, and then predicting on the held-out images. And this is kind of what we got, similar to the previous plot that I showed for the macaque monkeys. Again, we are showing you predicted and measured responses in one subject, and the left hemisphere and the right hemisphere. And these different colors, they correspond to whether these images are coming from the preferred category of stimulus for this area, which is FFA, or non-preferred. This is important because it's very difficult to actually predict how the brain would respond within different categories. So there's a lot of variance when you move across different categories of stimuli, but there's also variance that could be explained inside each of these categories. And as you can see, the model is relatively successful at predicting both of these types of variances. And these results were replicated in different subjects, and we also showed that that the model actually generalizes from subject to subjects too. But another interesting experiment that we did was on the behavior and comparing the accuracy of the predictions that the ANN model makes in reference to what we call the naive or M-Turkers, naive humans, and the human experts in these areas. For example, researchers that have been publishing in neuroscientific research articles on the same areas that we were trying to predict. And we had about 10 experts for these experiments and about 100 naive humans in this experiment. So for the naive humans, we basically showed them a bunch of images. We asked how face-like these images were, for example, for FFA region. And for the expert, we just asked them to predict how the FFA would respond to each individual image. And the results were, in general, very interesting. We found out that in most cases, when we actually looked at the images of the faces, the model was quite precise at predicting those responses, even compared to the human experts in the field. And when we looked at the subcategories of stimuli that we were showing, we found out that this also generalizes to other categories of images. And for some categories of images, like objects in this case, the model was actually much more precise than both naive humans and human experts. So this kind of tells us that the ANN model was better than or equal than humans, experts, at predicting the FFA responses. So as I said, we also looked at other areas, including EBA and PPA, which are known to be responsive or selective for body parts and scenes. And this was kind of the same story, maybe in some cases even better for EBA and PPA, that the model was even more precise than the experts and the naive humans. Yes, and ANN is generally more accurate at predicting responses within stimulus categories. So this was kind of all I had, just maybe one bonus slide that I wanted to show. So we also tried to use the predictive model in the same sense that we did it in cat monkeys to kind of like generate these synthetic images for each of these areas. And we still haven't tested these images to see how much they drive the neural responses in the brain, but to just give you a bit of feel of like what comes out. It's kind of, in most cases, it's very intuitive that for face areas, you actually end up getting faces out of this synthesis procedure. For body parts, we get things that kind of look, in some cases, look like complete body parts. In some cases, it's like complex, everything is together. And for PPA, things that kind of looks like scenes. So I just want to thank all my mentors and collaborators, especially Jim DiCarlo, Nancy, and Cordy Carr, who helped me a lot during this process, and I couldn't have done this without them. And finally, I just want to let you know that I recently started my own lab at McGill University. So if anything, any one of these slides or any of these projects is of interest to you, please get in touch. And I leave you with this summary slide.
All right, thank you very much, Pouya.
Finally, I just need to know that I wrote something because I hear back my way. So if anything,
I think it might be a good thing to close your second window if possible.
Yeah.
So thank you very much for the fascinating talk. This was absolutely amazing. I just remember when this came out in Science last year, I just was floored by that project. So there's a few questions that as you might imagine. So let's jump into them. First of all, first question by Abhilash. Why do CNNs mainly use only V1 architecture? Why not, for example, the rich diversity of PFs in prefrontal cortex, right? So, or more generally, right? Like, do you foresee doing something similar with other types of architecture in other brain areas? Right?
Yes, there is already ongoing work from Jim DiCarlo's group. That mainly I know about some work done by Cordy Carr, who's exploring these kind of ideas to use recurrent neural networks in combination with CNNs to kind of go a step more forward to additionally see what is the role of PFC in visual object recognition, and whether any of these models that are trained towards object recognition with a recurrent head, for example, could they be good approximation of what the PFC neurons, for example, might be doing in visual object recognition. My own lab also is focused on trying to push this more towards memory, in particular short-term memory, visual short-term memory. And the idea is kind of very similar to look at these recurrent convolutional neural networks and explore the different objectives, different architectures that might prove to be helpful in predicting those responses, not only along the ventral pathway, but also in prefrontal cortex,
right.
which is engaged usually during short-term memory.
Very cool. All right, let's move on to the next question. This one is from Mata Ramezanyan. I'm sorry if I didn't pronounce that correctly. Would it be worthwhile to study the neural signals coming directly from the retina instead of the representation in the visual cortex, since in the hierarchy the retina is closer to the visual stimuli?
It depends on the goal. So the retinal ganglion cell responses are still, I would say those are still some form of representations, right? They're shallower forms of representations. And we have different representations at different stages, of course. Of course, if you wanna understand what the RGCs or the retinal ganglion cells are doing, we should be studying those responses. But also, you might have a goal of knowing what this whole hierarchy is doing, and that's kind of what this work that I've been doing is mostly touching on, like what the system is doing and what's happening at each stage and what are the representations at each stage. So it's equally important to also look at RGC responses.
That's right. Okay, that sounds great. So next question. This one is from Sean Carreri. Can you associate the activations in specific layers of CNNs to different areas of the brain? Right. So this is seems like that's something that you are doing in your
right. Yeah. Right. So that's exactly what we have been trying to do. We did it to some degree, but for example, in the first study that I told you about, in the macaque monkeys, so the first thing that I did not discuss was to find out which layer in our architecture, which was a version of AlexNet, was the best predictor of responses in V4. You can do the same thing for different areas. We have done it for IT cortex. Others have done it for V1 and V2 cortices. And usually find that deeper layers are corresponding to higher cortices of the ventral pathway, like IT cortex. V4 area usually falls somewhere in between. And V1 and V2 are earlier, shallower layers of the CNN. So that's definitely something that could be tried. I just didn't have the time to discuss them in detail.
Okay, fair enough. All right, I think we'll have time for one last question, and this one is from Yann Charret, speaker from yesterday. So very impressive stuff. I was wondering how much influence the image set used in initial training of the ANN had in generating the novel images that stretched through the responses. Would different training images set influence the quality of the synthesized images?
So very good question. We have done this in simulation to some degree. We haven't tested the results though, meaning that we haven't closed the loop with different versions of these images, but we have tried using different seeds, for example, when we are creating the mapping functions.
Right.
And perceptually, all of these images, they kind of look similar, but they're not exact. In some cases, they turn out to be noisier. In some cases, they turn out to be more precise, less noisy. But in general terms, if I want to just describe them, if you have enough number of images, and enough is an unknown variable of its own, but if you assume that if you have enough images, usually you get images that are perceptually similar. That's the best thing I can describe.
So from that point of view on the variability based on the seed, like how big of a variability these different seeds lead in terms of responses, right? Do you have similar responses, or in other words, did you cherry pick some of the results to show us, or?
This was one of the, so let me go back to this. So
That's
this
right.
was
Oh yes, talked about
this
this.
was not a cherry picked. So at least this was among. So I don't have this here, but in the paper we actually show where this falls. I think this falls somewhere here, like along close to the median of the population that we had for all neurons. We had much better than this also, and we had I think about 20-30% of the neurons that we could not drive them as high as the best natural images. But this is kind of like, what we're saying is that if you do this over a population of neurons or neural sites, this is what you can expect out of this model that we did test. Of course, with improved models, we're hoping that this would also get improved. We just didn't have the time and everything to test all the models.
Right. All right. Thank you very much. So I think that's going to be it in terms of time. Please, well, thank you again for the wonderful talk, and please stick around here for the next talk, which will begin just shortly after a little switch period. So thanks again, Pouya.
Thanks, Gil.